Welcome to the first complex systems seminar of this year, and we're very happy that we have Aneta Koseska here from Bonn. So she is leading the Lise Meitner group at the Max Planck Institute for Neurobiology of Behavior, and is working a lot on transient dynamics, which, even though your application is more on the biological cellular side, is also very interesting for us in doing climate research, because we have transients as well. And so maybe regardless of what your field is, you can also take something away from this. So yeah, looking forward to your talk.
Thank you so much, for the invitation and for the fun introduction. And as Rai said, the basis of our research is understanding how cells compute and learn. But in order to do so, we identified some theoretical principles, which we also think are more broader because they rely on long transients that are guided by both scaffolds, and we have identified them in a large range of systems. So I would like to show you a little bit of how we think about this problem in general. So of course, complex systems are everywhere. We are focused more on the left side of this complexity. We study the dynamics of protein interaction networks in single cells, or also the neuronal dynamics when animals perform specific behavior. But then we have, on the other scale of the organization, a very similar from theoretical description of complex systems, different food webs or climate system that are also studied as a complex system. And the general question across all these very different systems is how does the complex dynamics emerges from the underlying network. And the classical theoretical approach for doing so, and I will demonstrate initially only on the side where we are interested in, and this is the protein interaction networks or neuronal networks. Basically, you have a network of interacting components, and then we have a system of differential equation that describes the eigendynamics of the nodes plus how these nodes are connected. And based on the autonomous properties of this system, we can use mathematical tools such as classical linear stability analysis in order to identify what are the stable solutions of the systems. So, as you are all familiar, probably we can have this dynamical description of the system also presented through a geometric description as a quasi-potential landscape, where each of these wells represents stable steady states, or attractors, and they are separated by unstable fixed points or cells. But what is the classical notion in analyzing complex systems is always we tend to identify the stable solutions of the system. So basically, also the mathematical tools are developed in order to identify the long-term or asymptotic behavior of systems. However, when we look in all these systems, none of them exist in isolation. A single cell, even a single cell in our body, they are continuously faced with many signals that come from the neighboring cells. This is an example of a neutrophil, which is a white blood cell that clears our organisms from bacteria. So the neutrophil is sensing a chemical that the bacteria is secreting in order to be able to chase it and identify it and destroy it, but also the neighboring red blood cells are secreting many different signals. So the biochemical network inside this cell, the neutrophil has to deal or is continuously forced with signals that change over time and space. The same is true also for the neuronal networks or the brains. Any brain does not work in isolation, but we are all continuously facing and have to interpret many different signals. So the rats, they don't live in the lab where they don't see any environment, but rather in reality, like this example, they have to face also challenges which are unexpected to them, as the snake coming out and the neuronal network of the mouse has to integrate many different tactile or visual or olfactory signals in order to be able to avoid this danger. This means that all these complex systems, they operate in environments or in worlds which are metastable, which are dynamic, and where many different surprising events have to occur, which means that they not only need to be stable, so the networks that process this information do not only need to be stable and robust and reproducible, but they also need to be able to adapt or flexibly respond to dynamic signals. But also, for example, for biochemical and neuronal networks in particular, it's important that they have a form of a memory so they can be able to integrate the signals from the past, make predictions, and be able to learn. So the very basic question that we ask is whether this attractor-based description or just identifying the stable states of the complex systems on the side of a chemical network or neuronal network is sufficient to explain the dynamics of this living system. So can we, using this formalism, can we understand how this systems operate when their surrounding or when their environment is dynamic, meaning when these networks or complex systems are continuously forced with a dynamic signal. And I would like to show you this in one example that we studied in the lab, which is related to the video of the neutrophil I showed. And this is, we wanted to understand how cells navigate in complex tissues. So when you have, for example, during embryogenesis or during wound healing, you have a cell that has to migrate, for example, from one side to the side of the wound in order to close the wound. And in order to do so, the neighboring cells, they secrete chemical signals. They act as a signal for the cell in order to guide it in which direction it should migrate. But these signals are not static and constant, and they are dynamic. They change over time and space. They are also noisy. So the biochemical network in the single cells is continuously forced with these dynamic signals to which it needs to respond. And just some basic biology, when you have a biochemical network in a cell that is faced with these signals that are usually coming from one side, what happens is that the proteins on the cell surface activate, they activate the signaling network in the cell. This activates the cytoskeleton of the cell, changes the shape of the cell into a polarized cell shape, and then the cell starts to migrate in the direction of the signal. So we studied a particular protein interaction network because we also do this experimentally, and I will show you some very minimal examples. But in general, we can look at this as any form of a network where now the nodes are the proteins and they interact with each other with specific network topology. And then we can analyze the dynamics of this network by understanding the number and stability of the different steady states. And we identified experimentally this minimal network module that regulates the dynamics of these surface proteins that are the ones that sense the signal and then drive the migration of the cell. And if we look at the topology, there is a double negative feedback there, so it's clear that this network has a form of bistability. That means that for specific concentrations of the protein on the plasma membrane, the dynamics of the biochemical network can be in two states. One is the basal or non-active state, and one will be the active state. Or if we talk in terms of cellular migration, then we have a cell which is symmetric and it's not activated. But then if we put a signal from one side, then this will activate the proteins on that side. This will polarize the cell, so the cell shape will also change, and the cell will start migrating. So if this cell biochemical network in the cell is organized exactly in this bistable regime, then when we present the signals to the cell, this will switch the system from the attractor of the basal to the attractor of the polarized cell shape, and the cell will start moving. This is, of course, very, very easy to predict, but the question to us was, how would the cell behave when the signal is not static, but when the signal changes over time and space? And just to come back to our quasi-potential landscape picture, this corresponds, of course, to a quasi-potential that has two wells, and these are the two attractors. One is of the basal, one is of the polarized state, and then the signal will switch the system from one to the other one. So we generated a physical model of the cell. You can model the cell as a viscoelastic system, where then the signaling or the signal from outside is going to change these viscoelastic properties. The cell will change the shape from basal to polarized, and the cell will start migrating. So in order to come closer to this migration scenario, in the simulation, we face the cell first with a signal from one side. So in theory, right, the system, when the cell is presented with the signal, the system will switch from this to the other attractor. The cell will start moving. But then what we did is also we interrupted the signal, we reestablished it again, we interrupted it, and we established it from another side, just to mimic these conditions that the signals in the environment are not constant, and to see whether switching from the basal to the polarized attractor will enable the cell to integrate and adapt flexibly to these changes of the signal in the environment. So in the simulations, if the system is positioned in this bistable regime, when we present the cell with the signal, it polarizes, it starts moving, but because it is now stuck in this attractor, of course, it has a memory of where it has seen the first signal and continues to move in that direction. But when we present it with a signal from an opposite side, the system cannot get out of this attractor. So already the first signal is sufficient to switch from the one to the other well in this quasi-potential landscape. But then if the signal changes, the cell is not able to sense this. And this, of course, is not useful for the cell, meaning that the cell is not able to sense signals which change over time. So from a mathematical aspect, this tells to us that the attractor-based framework is potentially not suited to describe how systems respond to signals that change over time. So we tested whether this is true experimentally. So we have epithelial cells, which we put in microfluidic devices where we can generate these graded signals. So we generate the graded signals, we change the stiffness of these signals, or we make the signal dynamic. Then we disrupt it, we reestablish it again, we switch the direction, and then we see whether cells are capable of solving this problem. The signal is from the left, the cell polarizes, starts migrating in that direction. It doesn't stop when you remove the signal, so it has a certain memory where it has seen the signal, so it continues in that direction. And when it loses that memory, it goes into a random walk migration. And I just realized that I didn't put the other video here, which shows that when we change the signals in the environment, the cell is completely capable of integrating these signals. But one important thing to notice here is that the green line is where it stops when we switch off the signal. Then you can see that the cell still has a memory and continues migrating in the same direction. But this, of course, if this would be a stable attractor, then the cell will migrate in the same direction forever. But here we see that this is temporal, and in these cells, it's 40 to 50 minutes. So how can this be? Where is this memory coming from if it's not from a stable attractor? So what we could show also experimentally, but also from theoretical, this is the only possibility, that the biochemical network is not organized in this bistable regime, but it's actually sitting very close to the bifurcation. And because it's sitting very close to the bifurcation, there are specific properties that the system inherits. But then you might ask, well, if I'm very close to the bifurcation, I have only one single steady state, and this steady state is of the basal activity or of the unpolarized steady state. So how does the cell polarize, and how does it have a memory when we switch off the signal? So let me unpack this a bit in detail. The biochemical network that we studied also experimentally, so we have very detailed measurements, so we can model this in detail, and we can also not just do the bifurcation analysis, but also calculate these quasi-potential landscapes and thereby understand how the system behaved in the presence and absence of a signal. So when the system is organized close to the saddle node bifurcation, so close to the bistable regime, the quasi-potential landscape has only one well, which is the basal unpolarized state. However, because it's very close to the bistable system, on the other side, this landscape is not a well, but it looks rather like a chair, which is slightly tilted. If you push the system a bit more in the bistable regime, this part will also become a well, because this is where the second steady state will happen. So what happens when we give a signal to the system? This landscape is not fixed anymore. So our picture of an autonomous system where we have a fixed landscape and where the signal just switches the system between landscape is not completely correct, and especially not correct when these signals change over time. So when we give a signal to the system generates a new steady state, and this is a monostable regime. This is the active state. When we remove this signal, we come back to our initial quasi-potential landscape, but now the system falls onto this rather flat part of the quasi-potential landscape. So this quasi-potential landscapes looks a bit like this. You come down from three sides, and then you land on the seat of the chair, and because it's slightly tilted, the system will slow down, which in our case is what gives the system a memory of the active state, but eventually you will fall off. So this is not a classical stable steady state, but it's what in the literature is known as a ghost set, because it's a ghost of the steady state that existed before the landscape has changed. So this type of ghost sets are not stable steady states. They, however, give the system a slow timescale and a certain form of a quasi-stability. So in the case of the biochemical network that we studied, the timescale of the ghost set is an order of magnitude longer than the intrinsic timescale of the biochemical network. So the system gains a slow timescale for free just by sitting close to this bifurcation. It gives the system a quasi-stability with this, because the system can maintain, for example, the cell can maintain still to migrate in certain direction for a given period of time. But the system is still flexible how it responds to other signal, because this is not an attractor, and it's easy for the system to respond to the new signals. But this also means that in this case, we're not dealing with asymptotic dynamics of the system that goes to an attractor, but rather what is important for the biochemical networks in this case is that they actually make use of this non-asymptotic dynamics that comes from these long transient states. And more importantly, what we also see here is that we go away from this picture of a fixed landscape, but rather when we have a complex system that is forced with signals that change over time and space, it looks more like the picture on the right, where you have a landscape with strings where you're pulling down this landscape. So this means that the mathematical framework is in this case a non-autonomous description of the system. And this is an important distinction also mathematically, because for non-autonomous systems, both the topology or the geometry of the landscape can change, meaning that both the number and stability of steady states can change, but also their shape and the size and the proportion of the attractors. So if these are not any more fixed characteristics of the system, we can, just by characterizing the number of attractors in the system, is not sufficient to explain the dynamics of the system. For example, the memory that we have seen in the cellular migration. We cannot explain by the presence of the attractors, but even mathematically in this case, the description or the solution of the system will be the trajectory in the state space, and that trajectory in state space is in this case guided by these ghost-like sets or these objects in state space that are quasi-stable. So we did some more detailed mathematical characterization of this ghost sets. And the most simple example is with a normal form of a saddle node bifurcation. So when the system is when the system is smaller than 0, you have one fixed point and a saddle. When the alpha is larger than 0, or there is no fixed point in the system, but you have this ghost set when alpha approaches 0 from the positive side. So we wanted to compare the properties of the ghost set with the dynamical properties of a saddle, because a saddle is an unstable phase-space object, and our ghosts are also not classically stable in a Lyapunov sense. So we took the comparison to the saddles. In order to be able to identify the characteristics of the ghost set, we constructed a scalar function, which in this case is something similar to the kinetic energy of the system. And you can, even for systems where you cannot do analytical analysis, one can calculate this quantity numerically. So when you minimize this, for example, for a fixed point, Q will be exactly 0, which we see exactly for the saddle. However, for the ghost, Q is close to 0 in a large region in phase space. And this corresponds to the flatness of this region, and this defines how large the ghost set is in the system. So now, because our kinetic energy is close to 0, we can also locally linearize the system and calculate locally the maximal or the eigenvalues of the system. So if we look at the maximal eigenvalue for the saddle is positive, which says that this object is unstable. But for a ghost, we can see that the maximal eigenvalue goes from negative by 0 to positive, and having the maximal eigenvalue also around 0, this additionally explains why we get the slowness or the additional slow timescale when the system falls into a ghost set. But this also explains how the trajectories are guided, or how the ghost guides the flows of the trajectory in the state space. So in the saddle, of course, the trajectory comes along the stable and escapes the system very quickly along the unstable manifold. But in the case when the system has a ghost, everything falls down on the ghost set, then evolves very slowly along the ghost, and then it escapes. So we also then next tested how stable the dynamics will be on a saddle versus on a ghost when there is a noise in the system. So we calculated the total trapping time on a saddle and on a ghost system when we increase the noise intensity. And for a saddle, this is well known that as you increase the noise intensity, the total trapping time will exponentially decay, because you have two unstable directions where the system can escape the saddle. But for a ghost on the left, we see that as we increase the noise intensity to rather high noise values, the total trapping time stays robust to the noise. And this is because the system has only one direction where it can escape, so it can tolerate also very large noise intensities, which means that for natural systems, such as the biochemical networks that we look at, which are inherently noisy, it can also explain how these biochemical networks can also use such unstable objects in order to guide the dynamics of the cells. So in contrast to the classical framework, where we always look only at the stable steady states, in order to characterize the dynamics of the system. Here we see that even an instability or a quasi-stable object in a phase space can be of advantage because it gives the system the both sides, the transient stability and also the flexibility. So for engineered system, of course, stability is primary, but natural systems, because they operate in a very dynamic world, it is necessary to balance both the stable side, but also to be flexible when the dynamics of the environment changes, and therefore these unstable or quasi-stable objects are likely better in order to guide the dynamics of the system of natural systems. So are these long transients that we observed from ghost sets just one-time finding of this special biochemical network that we studied, or is transient dynamics important in many different systems? So when one looks in the literature, there are many reports about transient dynamics, and it has been discussed why transient phenomena or long-lived transients are actually more important in ecological models than looking only at the steady states of the system, but not only in ecological models. They are also very important to describe the dynamics of neuronal networks. And I would like to show you here one example. The study is now rather more than 15 years old. It was an experimental study where they wanted to determine how the brain of a rat processes or encodes different tastes. So what they did is they presented to the rat different tastes, such as sucrose, quinine, and so on, and then they recorded the neuronal dynamics in the rat gustatory cortex. So what they observed is that for each of the different tastes, how the system responds, it encodes this taste into a sequential switching dynamic. So you have a neuronal firing pattern that is transient, and that from one it goes to another firing pattern, and so on. And for each of the tastes, there was a different sequence of firing patterns. If you would repeat the same taste again, the firing pattern does not change. Just when the second sequence in the pattern would happen might vary. So it was clear that the tastes are encoded not through the neuronal network going to a specific steady states, but rather through these sequential switching dynamics of transient firing patterns of the neuronal networks. So this is one example. In the meantime, there are a range of different examples showing that in many different instances, in particular in olfactory or motor control, the neuronal networks is actually processing this information by using these long transients, and very often these long transients are presented in such sequential switching manner. And description of such transient dynamics where the system switches from one long transient to another long transient is also not new to theory. The first proposal, at least to my knowledge, has been the description of sequential switching dynamics in chaotic systems, or the chaotic itinerancy, with the work of Tsuda, Ikeda, and Kaneko, where the system goes into one of a chaotic attractor, and stays there for a relatively long time, but then it can escape, switch to the next one, and so on, and you can do this switching in an ordered sequence, depending on the dynamics of the system. And this was initially proposed to describe exactly this type of observation in neuronal networks, where you have sequences or switching between sequences of neuronal firing dynamics, and to go away from the chaotic description, because not always chaotic dynamics has been observed in experimental measurements of the neuronal dynamics, and in particular to explain such sequential switching dynamics for olfactory processing, the heteroclinic objects have been proposed, first identified in ecological models, but then the framework has been developed by Rabinovich, Theogeisel, and further on. And these heteroclinic objects are phase-space objects where you have one saddle, where the unstable direction of the first saddle becomes a stable direction of the second saddle. Then the unstable direction of the second saddle in the sequence becomes the stable direction of the third saddle. So you are just ordering the saddles in the phase space. So then the system first spends some time on one saddle, then it will switch and spend some time on the second saddle, switch, spend some time in the third saddle. So you can get long transient because the system stays on the saddle, but then it can switch to the next saddle in the sequence. And in analogy to this type of objects, after we define the ghost-like sets, and we have also formal mathematical prescriptions, it was clear that also one can find such objects of connected ghost in phase space. Because then you take the unstable direction of the ghost, which becomes a stable direction for the next ghost. So the system falls on the first ghost. It will spend long time, so will generate this long transient here, but then it will switch and go to the next ghost in the sequence and so on. So we wanted to, as we did the comparison of the properties of the single ghost versus a single saddle, we also analyzed the properties of a ghost channel versus a heteroclinic channel. So you can generate these as a very basic geometric objects, and then we looked how the properties of the dynamics correspond when we have noise in the system. And this is very similar to the dynamics with the trapping time that we calculated in a single ghost versus the saddle, that saddles are unstable with respect to noise, which you can see also on the right side for a heteroclinic channel. Then when you increase the noise intensity, the trajectory very easily can escape and not stay on the heteroclinic in the heteroclinic channel. And this is not the case for a ghost channel, because there is only one direction where the dynamics can be guided. Basically, this enables to guide the flows in phase space, and this is robust to the noise intensity, which we can quantify. In this case, we quantify the Euclidean distance between the trajectories in the heteroclinic and the ghost channel when we increase the noise intensity, and we can see that in the ghost channel, even when you increase the noise intensity, the trajectory is still well aligned in the channel, and this is not the case for the heteroclinic channel, which suggests that natural systems would likely use, or the dynamics of natural system would be likely described by ghosts rather than heteroclinic scaffolds, as the ghost scaffolds are more stable with respect to noise. So to come back to the example I mentioned of this cyclic switching behavior that has been observed for the encoding of the taste, it's also possible to arrange the ghosts or the saddles not on a line but in a cycle. So as you can generate heteroclinic cycles, we can also generate ghost cycles. And then you can see that the system first is on one of the ghosts. It gets the transient stability by staying on this ghost, but then it switches to the next one in the sequence and so on. And this stable switching pattern is not destroyed when we increase the noise in the system, which could be a mechanism how this sequential switching dynamics observed in neuronal systems can be generated. In contrast, if you would compare these to a heteroclinic cycle, we can see already from the time series that when we increase the noise intensity in the heteroclinic cycle, what we do is we reduce the trapping time on the saddle. So we don't have this good separation of a slow positioning of the system on the saddle versus this fast switching between the saddles. And you can see this also in this video, that on the left for the ghost cycle, the trajectory very nicely follows and switches between the ghosts while spending a long time on the ghost. Whereas for the heteroclinic cycle, it's very easy when you introduce even small amounts of noise that the trajectory escapes the heteroclinic cycle. But also, if we quantify the period that the system has on the ghost cycle or the heteroclinic cycle, the trapping time in the vicinity of the ghost, and how long the system spends switching between different ghosts and different saddles. This is very similar to the dynamics on a single ghost. We see that the period that the system has in the ghost cycle stays constant, even when we increase the noise intensity to intermediate levels. The trapping in a ghost vicinity stays constant, and we still have a very good separation between how long the system is trapped on the ghost versus fast switching between the ghosts. But when you do this for the heteroclinic cycle, you can see that already at intermediate noise intensity, the system spends more time switching than being trapped on the saddle. So if you would like to encode information in such a sequential switching-like activity where you have a long transients and then you switch to another long transients and so on, then heteroclinic cycles cannot explain this type of dynamics, because this type of dynamics is very easily destroyed on a heteroclinic cycle when there is noise present in the system. So we studied these properties of these ghost cycles. How do they respond to dynamic signals, in particular when they're forced with signals with a specific frequency? So we generated. Actually, it's a modification of the Van-der-Waals system, but maybe the details not so important. It's a system that has, these are the nuclines of the system, and the system when here and here the nuclines are very close, the system has two ghosts, so you can get something that looks like a fast slow oscillations, but only emerging from a ghost cycle. This means that the system stays in the one ghost, then it switches to the next ghost, it comes back, and so on. And it looks very much from the time series as any fast slow system. However, ghost cycles have very different properties when they are forced with a signal with different frequencies. Because in the case of the ghost cycle, as a typical also for many non-autonomous systems, is that the landscape of the system changes. If you can look, so the green here is the forcing from the outside, and you can see as the system goes through the forcing, these nuclines, they change their position. So the nuclines spread apart, then they intersect, and so on. And because of this, maybe I play the video once again. I create and destroy fixed points. I get this ghost, so the system gets trapped there. It gets released with the forcing. It goes to the other side. It's going to get trapped there, and so on. And this is very similar to a rate-dependent tipping in a non-autonomous system. And actually, one can analyze, and we have analyzed the system in such a way. But what you can see is that the system can entrain to a very broad range of frequencies into one-to-one manner. Classically, when you calculate the entrainment of any limit cycle to different forcing frequencies, you have very narrow and very defined Arnold tongues. But in this case, because the landscape of the system changes, the system can entrain with one-to-one for a broad range of frequencies. As two examples, they are order of magnitude difference to the intrinsic frequency of the system. So this, to us, it suggests that a natural system that would operate with this long transient dynamics that can be structured, for example, as a fast slow system, is a very flexible system that can very naturally adapt to the changes in the environment. Such a richness of the dynamics of this of systems that have this long transients, which are guided by ghost scaffolds, being channels or cycles, and made us to think how common are these ghost scaffolds? Can we find them also in different systems and outside of biology? And we looked in one climate model of cascade tipping that comes exactly from here, and that is the model of the coupled North Atlantic Ocean and the El Niño oscillation system. And actually, with just a very narrow or small reparameterization of the system displays this cascade tipping can be explained as a system going through a channel of ghosts, where you get the system stays in a long transient for a relatively long period of time, but then it can tip and goes to the next long transient and so on. However, whether this tipping is the system crossing the bifurcation or the tipping comes because the system is in a long ghost and then just switches to another ghost has many implications of how we study tipping points in ecological or climate systems. So one part of the work, what we are trying to do is try to identify a characteristic of these long transients from experimental data and whether we can have some indicators how long the system stay in this ghost and if we can predict when the system would exit this long transient behavior. And just to summarize, we have described this emergence of quasi-stability in a range of natural systems that comes from these ghost scaffolds that I showed can be arranged. Also, you can have more larger phase-space structures of ghosts, so the system can switch between different ghosts in a sequence and generate this cascade switching behavior. They can be also combined with saddles. So basically, you can have very complex scaffolds in phase space that generate these long transients. But what is important to contrast to the classical picture is that they are not captured by an attractor because these sets are not Lyapunov stable, but they evolve non-asymptotically under the influence of ghosts, which means that we also need different tools how to analyze and how to describe the system, but also how to identify them in experimental data. And especially for natural systems, we think this is important because not only it gives robustness to the system, where the system can maintain its dynamics for periods of time, which are, in the case, for example, of the cell, magnitude order longer than the underlying dynamics of the network. But more importantly, it enables the system to be flexible when it is faced or the system is forced with dynamic signals in the environment. So it allows this natural balancing between robustness and flexibility of the system. So with that, I would like to thank the people who did the work. The first part on the biochemical network and cell migration was done by Achilles Navishev, and then Achilles, Daniel, and Gayatri did the theoretical characterization of the ghost sets in collaboration with Ivan Tukin and Alexander Gorban and Ulrike Feudel. And I would like to thank you for your attention.
Thank you very much. Great on time as well. Any questions for Aneta? Maybe I will start off while you think of some. So when you're comparing the ghost states and the saddles, do you make any restrictions on the curvature locally? Because of course you could continually move from one to the other, right? So when you quantify these,
I'm not sure if one could continually move from one to the other, but what we ensure is that the trapping time in absence of noise is equivalent both on a saddle and on a ghost. And then we see how this changes when we introduce noise to the system.
Okay. And then, yeah, okay.
So if I understand correctly, biological and ecological systems naturally sit at the bifurcation, or like in a critical zone, very close to bifurcation, right?
This is the example that we identified. I can name, and we have some confidence of it from the experimental work. I can name several other biological examples that have been experimentally identified that sit close to the bifurcation. But for example, and I can say this, for example, for the cell biology literature, but these are still a handful of examples. I think both the experimental methods, but also the theoretical description so far has not been right to look for these possibilities. My guess is for natural systems that this would be an optimal point to sit, exactly because of this balance between robustness and flexibility, but that would be a speculation.
And do ghosts, I mean, is it a universal feature of some types of bifurcation?
Yes, yes. So for saddle nodes, definitely, and you can have it even if you have a saddle node of a subcritical, how subcritical half of pitchfork is stabilized and so on. Just then you have a ghost of a different of the attractor that has existed. We are trying, or well, not we, but rather our collaborators, even in Alexander, who are mathematician, they have extended the Lyapunov framework in order to be able to define these both sets generically. So still, most of our examples come from the system sitting close to specific bifurcation or SNCC and so on. Whether there is a different way how to identify and how to define them, I think it's an open question.
Is it possible to use this information, knowledge, maybe even in medicine somehow, that the ghosts are in some way more effective, if I understand your presentation correctly, in the sense that they behave longer in the saddle points than with the other method. So maybe the speculation,
yes,
but I mean,
what we are doing is basic research. But for example, examples that we are also looking at that could potentially have some applications is of course the epithelial to mesenchymal transition in cancer cells. So how cancer cells start let's say migrating or metastasizing, because having a memory on the level of the biochemical network would be important in many examples. It would be important in regeneration. It would be important in cancer treatments, and this is the area where we are looking at. I mean, also during embryogenesis as well, because in many cases, right, if you have perturbations to the system, embryos are still robust to certain number of perturbations, and they can recover on their own. So many of these processes could be potentially guided by such a molecular memory. But it's very far from any application. This is just a play system. Yeah.
I have a question for my own understanding. It's about cell migration, because I know a cell pulse model and to simulate cell elongation, but because this ghost scaffold, this is the first time for me to hear it, so it's very new for me. So this method can also to simulate a cell migration, how cell moves is also kind of.
So this is a bit different, or what we have done is a bit different than the cell pods model. So we model the cell basically as an electronic circuits, as a weight system, and we model the shape of the cell as a zero level set of a potential function. And then we coupled that, we coupled that to a partial differential equation model that describes the dynamics of the biochemical network that guides. So then the biochemical network tells the system how it should contract and what is the speed of migration and so on. So this is rather a physical model for the cell. I'm not sure if there's any way to introduce this type of dynamics to cell bots models.
Okay.
So this is a more detailed physical model of the cell because we wanted to come closer to the experimental work. Thanks.
You compared the ghost sets to the heteroclinic objects. Did you also do the comparison with the chaotic attractors? With the chaotic attractors?
No.
Is there a reason?
Because it would be, I think, for us at the moment, very hard to completely characterize the dynamics of the chaotic sets. So I wouldn't know even which property to compare. But this is the only reason.
I was thinking about this as well when you introduce this kinetic energy measure that also becomes difficult when you have chaos, right? Because then you need to decide in which direction you are measuring your energy.
So we restrict, we stayed consciously out of the chaotic dynamics, but yes, this will be something which is important to think about.
All right, any final question? If not, then thank you very much, Aneta. Very interesting talk.